Okay, so the next part of our notes is we're going to look at how Gregor Mendel determined these traits. Well, he determined these 7 distinct traits of these pea plants by performing what are called crosses, genetic crosses. A cross, what that is a fertilization event. So it's mom plus dad equals kids. And we use Punnett squares so that we can look at all of the possibilities in those kids. So what you see here on this little example that I just found from the internet shows you how these crosses work with a Punnett square. So this Punnett square, what it does, it helps us look at all possibilities of connecting or fusing or uniting the genes for a trait that dad has to provide. We can see those on the left, and those that mom has to provide, and we can see those on the top. Now, if you notice, there are 2 copies. So if you remember, everybody has 2 copies of every gene because everybody has 2 parents. So this is how Mendel started, and let's go ahead and click here. He started with what are called self-pollinating experiments. So self-pollinating plants, like these pea plants, they produce offspring identical to themselves. Now, the reason why is, and if you've probably seen these before, is that these flowers, these flowering plants, have the male, the stamen, the anther, and the filament. So that's the male part of this plant. And the female part over here, the carpel, the stigma, the style, and the ovary. So they have male and female parts of the plant, which produces their own gametes. So that means that this plant can self-fertilize. But when we're talking about plants, we call it pollinate. So it can self-pollinate. And you can see this again on the right side. So if you leave these plants alone, the pollen, which is the male gamete, can come from, and you can see 2 different versions of these, the anther. It's the top of the filament. It can come from this area of the anther and then attach to the female part, the carpel. Now, the carpel here is made up of 3 parts, the stigma, the style, and the ovary. The stigma, which is that top part right in the middle, it's kind of sticky, so any pollen that flies along is going to stick to it, and then it can travel down this little tube called the style, so it's going to protect the pollen and allow it to get down to the bottom part, which is the ovary. Now, inside of this ovary is an ovule. So that is the female gamete. So that's equivalent to a female egg if we were talking about a human. So if you have purple pea plants that are self-pollinating, you're going to make more purple. If you've got white pea plants that are self-pollinating, they're going to make more white. Same thing with all of the other 7 alleles. Now, this self-pollinating experiment, not a lot of experimenting going on with that. But one thing that does come from this, we call these organisms that, when allowed to self-pollinate, to make more of themselves, one term you might hear for them is true breeding. Now, some people use the word purebred. I kind of stay away from it, but true breeding. Now to the actual experimenting part of the experiment, cross-pollination. So in this case, this required a little bit of manipulation on Gregor Mendel's part, and you can see an illustration on the right showing you this manipulation. And this set of experiments, or in these set of experiments, what he did is that he took 2 different plants and he crossed them together. Now, it wasn't just like 2 different purple plants or 2 different tall plants or 2 different short plants. What he did is he took plants that had different forms of the trait. So he took a tall with a short, a purple with a white, ones that had pod shapes that were smooth and ones that had pod shapes that were bumpy. he put them together, and it did require manipulation. So over here to the right again is this illustration of the manipulation. And essentially what he did is that he went into a plant and he removed the part of the plant that produces the pollen. Now, the pollen, again, is equivalent to sperm in a human. So he removed that part so this plant could no longer self-pollinate. And we can see that he's doing that with this purple one. Then, just using a little, not a toothbrush, that's a paintbrush, he went over to the white and gathered pollen from the white flowered pea plant and dusted it onto the stigma, which is the female part, stigma style ovary of this purple plant. So this is cross-pollination. It did require some manipulation. So he didn't want to have a possibility that any of the genes from the purple plant got into that female part to pollinate that ovule. So he removed it. So another little piece of word art here to hopefully help you with remembering what's what. So cross-pollination. Cross-pollination, different from self-pollination, in which we take two different organisms that have different forms of a trait. So in this case, we saw purple, which is dominant, crossed with white, which is recessive. Or we can have tall, which is dominant, crossed with, short, which is recessive. And then what we end up getting is a hybrid. So that's something we haven't seen before.